Imagine you're at like an incredibly noisy, crowded party. You know the kind.
Oh yeah, absolute sensory overload.
Right. There's music blaring, glasses clinking, people are just shouting over each other, and someone is, I don't know, laughing right next to your ear. It is total chaos. But somehow, through all of that, you can still perfectly focus on one single conversation with the person standing right in front of you.
It really is a marvel of biological engineering when you stop to think about it, because our brains are constantly bombarded with a staggering amount of raw data from our environment. By all logical measures, the system should just crash.
Yeah, like a computer freezing up with too many tabs open.
Exactly, but it doesn't. Your brain somehow filters out the noise and amplifies the single signal you actually care about.
And to understand how we actually pull off that party trick, we are diving into some really groundbreaking research today. Welcome to the Deep Dive. For you listening, our mission today is to completely unpack a phenomenal 2026 paper published in the journal Neuron by Hain-Mueller and colleagues. We are going to map out the microscopic gatekeepers in the brain that make memory and focus possible, leaving literally no stone unturned.
It's a fantastic paper. We're looking at a specific region in the hippocampus, which is the brain's memory center, called the dentate gyrus.
The dentate gyrus, or DG, right?
Right. The DG is arguably the most critical bottleneck for memory formation. You can think of it as, well, the ultimate gateway. It's constantly receiving these massive streams of sensory information from another region called the entorhinal cortex.
And it has to decide in like a fraction of a millisecond what information gets to form a lasting memory and what gets discarded. I've always liked to think of the dentate gyrus as the most exclusive nightclub in the brain.
I love that analogy.
Right. So the sensory input's coming from the entorhinal cortex. That's the massive crowd of people lined up outside on the street. They're all shouting, trying to get in at once. But they can't just storm the doors. The club has bouncers.
And in the brain, these bouncers are a class of cells called interneurons. Today, we're focusing heavily on two very specific types of these bouncers: the parvalbumin, or PV interneurons, and the somatostatin, or SST interneurons.
PV and SST. But, I mean, the brain isn't just a neat little diagram. It's a dense microscopic soup of cells all tangled together.
Yeah.
So how did the researchers actually look inside the brain of a mouse that's physically running around a maze and say, that specific cell is a PV bouncer, and that one over there is an SST bouncer?
It's a great question because it's notoriously difficult. It required a combination of some of the most advanced techniques in neuroscience. But before we get to the tech, let's establish the full cast of characters in this nightclub.
Okay, lay it out for us.
So we have our excitatory cells. These are the patrons actually trying to pass the signal forward into the club. First, we have the granule cells, or GCs. These are the sparse, quiet types. Their main job is something called orthogonalization.
Orthogonalization, that's a big word.
It is. It essentially means they take similar inputs and separate them, ensuring you don't confuse two similar memories, like remembering where you parked today versus where you parked yesterday.
Okay. That makes sense.
Alongside them, we have Mossy cells, or MCs. These are much more active. They fire more often and act as amplifiers for the signal. So those are the patrons.
The quiet ones and the loud ones.
Exactly. And then we have our bouncers, the inhibitory cells, whose job is to pump the brakes. First up are the parvalbumin, the PV interneurons. These are incredibly fast spiking cells. They physically connect to the main body, the parasympathetic region of the excitatory cells, allowing them to apply extremely fast, powerful, immediate inhibition.
Like a bouncer grabbing someone by the shirt collar right at the door.
Perfect, yes. Second are the somatostatin, or SST interneurons. They wire themselves differently. They target the outer branches, the apical dendrites of the excitatory cells, so they provide a much slower feedback loop of inhibition.
So we have our fast bouncers and our slow bouncers. But again, how do you watch them in a living animal?
They used optotagging. Essentially, they took genetically modified PV-Cre and SST-Cre mice and used viral vectors like tiny molecular delivery trucks to insert light-sensitive proteins into just the specific bouncer cells.
Light-sensitive proteins, so they literally control the brain with lasers.
They do. They inserted two types of opsins: CHR2, which acts as a switch to excite the cell when exposed to blue light, and ENPHR, which shuts the cell down. By implanting a microscopic optical fiber and a tiny sensor probe, they could flash a laser and instantly see which cells reacted.
That is wild.
If a cell fired within 5 milliseconds of the light turning on, they had a confirmed identity tag.
But they didn't stop there, right? Because you can't just flash lasers the whole time the mouse is trying to learn.
Right, so they took the electrical data from those confirmed cells and trained a machine learning algorithm, a support vector machine, or SVM, to recognize them in the wild. They fed the algorithm the unique physiological features of each cell, specifically the action potential width and the shapes of their spiking autocorrelograms, or ACGs.
So basically the unique rhythm and shape of their electrical spikes.
Exactly. And the SVM algorithm was remarkably successful. It classified the granule cells, mossy cells, PVs, and SSTs with 90.06% accuracy.
90%, that's incredible. You know, a good way to visualize this for you listening, instead of getting bogged down in the complex wave graphs, think of it like a landscape. The granule cells are like a wide mountain with a distinct little shoulder on the slope. The mossy cells are a steep, narrow peak. And the PV and SST bouncers, they're highly regular. They just tick away like fast, reliable metronomes, with the SSTs ticking right in the middle between the PVs and the excitatory cells.
That's a really helpful visual mnemonic. And the algorithm picked up on those subtle differences to identify exactly who was talking in real time without needing the laser constantly.
Okay, let's take a quick breather and do a mini recap of our first subtopic here. Bullet 1. The dentate gyrus geek keeps memory like an exclusive nightclub. Bullet 2. Optogenetics uses light, specifically CHR2 and ENPHR, to tag and control specific cells. Bullet 3. An SVM machine learning algorithm successfully classified our 4 cell types based on their unique electrical signatures with over 90% accuracy.
Spot on.
So a quick question for you listening to mull over. If you were to design an algorithm to classify human behaviors based on quick snapshots, what features or waveforms would you track? Just something to think about.
Well, that's a great thought experiment.
All right, moving into part 2. We have this incredible technology to tell our fast and slow bouncers apart. We can watch the nightclub open for business. But I have to push back here because when I first read this part of the study, I was confused. If these inhibitory cells are just the brakes of the brain, why do they care where the mouse is standing? I thought they just shut things down.
It is highly counterintuitive. You'd assume a cell whose only job is to yell stop doesn't need a map of the room. But the researchers tested this by looking at spatial information, or SI.
Which is measured in bits per spike, right?
Yes. They measured how much information a single electrical spike tells us about the animal's physical location. They tracked the mice running on a linear track, then a radial 8-arm maze, and finally just wandering an open field.
And what did they find?
Well, the excitatory cells, the GCs and MCs, acted exactly as you'd expect. They had high spatial tuning. They act like highly precise GPS coordinates, firing mostly when the mouse is standing in one specific spot.
Right, but the inhibitory interneurons...
Generally, their spatial tuning was lower. They are the brakes. But the twist is that when they compared the 2 types of bouncers, the slower SST cells had significantly higher spatial tuning than the fast PV cells. The SST cells actually had a distinct sense of place.
Wait, really? Why would the slow bouncer have a better map of the room than the fast bouncer?
It comes down to a mechanism called synaptic convergence. The researchers mapped the brain's wiring using something called short time scale cross correlations, or CCGs, basically tracking which cells reliably fire back-to-back within milliseconds to prove who is directly plugged into who.
Okay.
And they found that the fast PV cells receive input from a massive, highly diverse pool of excitatory cells.
So it's like a newspaper editor who reads 50 different local beat reporter stories from all over the city and just has to write one vague, overarching headline.
That is exactly it. Because a single PV cell gets inputs from so many different cells firing in different physical locations, its spatial awareness averages out to a blur. It's just taking in the general vibe of the entire room.
The vibe check cell.
Exactly. But the SST cells receive much more specialized, less divergent inputs. They listen to fewer, more localized reporters, so they retain a much sharper focus on specific locations.
It's funny how that applies to our daily experience. You walk into a crowded, unfamiliar restaurant. Your inner PV cells are just registering the broad strokes. We're indoors, it's loud, tables everywhere. But your SST cells are locking onto the specific corner booth where your friend is waving at you.
And that division of labor is absolutely critical when the actual memories start trying to push their way through the door.
Okay, recap time for part 2. Bullet 1. Mice ran mazes to test spatial memory. Bullet 2. Excitatory cells act like precise GPS points. Bullet 3. SST interneurons have better spatial awareness than PV interneurons due to their specific, localized input wiring.
Perfect summary.
So, a question for the listener. Think about your own memory. Are you more like a PV cell, taking in the general vibe of a room, or an SST cell, focusing on the specific location of a few key details? Let's get into part 3. The dentate spikes. We know the entorhinal cortex is basically shouting at our dentate gyrus nightclub. How does the signal actually cross the threshold without overwhelming the system?
It doesn't happen continuously. It happens in bursts called dentate spikes, or DSs. These are synchronized bursts of energy and information from the entorhinal cortex. They're absolutely critical for memory consolidation.
Right.
What this study did was break down a single dentate spike, millisecond by millisecond, to see the exact chronological mechanism.
The play-by-play.
Exactly. So the memory burst hits. Instantly, before the peak of the spike even fully forms, the PV cells react. They fire early negative 1.5 milliseconds before the peak. They absorb the initial shock.
Then what?
Next, at positive 1.0 milliseconds, the excitatory granule and mossy cells finally fire. The memory information is actively passing through the gateway.
But then, coming in dead last, a full 1.5 milliseconds after the peak, the SST cells fire. Why are they arriving so late?
Because they act as a sluggish gatekeeper. They lack direct inputs from the entorhinal cortex and rely on feedback from the cells already inside the club. So they activate late by design. They allow a brief window for the information to pass, and then they act as a temporary stop sign, slamming the gate shut.
And the researchers proved this by hacking the system with optogenetics, right?
They did. When they used the EMPHR opsin to inhibit and silence the SST cells.
Basically turning the stop signs off.
Exactly. The floodgates opened. The overall rate of dentate spikes shot way up. But when they used CHR2 to stimulate the SST cells, forcing them to stay active, the dentate spikes plummeted to 23.94% of baseline.
Wow, they just locked the doors completely.
Yes, and interestingly, manipulating the fast PV cells had almost no effect on the overall rate of these spikes.
So, a great mnemonic for you listening. Remember that SST stands for stop sign temporarily. They are the final brake pedal on the memory signal. Let's recap part 3. Bullet 1. Dentate spikes are memory-forming bursts of data. Bullet 2. PV cells react instantly to absorb the shock. Bullet 3. SST cells react slowly, providing a specific window for data to enter before shutting the gate.
Spot on again.
Question for the audience. When you're trying to learn something complex, do you ever feel your own SST gate closing, where you just physically cannot take in any more information? Like reading the same paragraph 4 times. That's your SST gate protecting your brain.
It really is an elegant mechanism. But that brings us to the climax of the paper, part 4. The paradoxical excitation and the winner-take-all dynamics.
Yes. This part blew my mind because functionally it makes zero sense. I have to push back on the basic physics here. If you use a laser to stimulate an inhibitory cell, a cell whose biological purpose is to hit the brakes, the whole network should just go quiet. It's like a teacher yelling quiet in a classroom. Everyone stops talking. But the data show something highly paradoxical.
It completely defies simple intuition. When the researchers used 1-second light pulses to stimulate the PV or SST cells, yes, the vast majority of surrounding cells went quiet. But they observed that a small, select group of granule and mossy cells experienced a delayed, paradoxical increase in firing.
Wait, so if stomping on the brakes, the brain actually forced a few specific cells to hit the gas. How do we know this isn't just a glitch in the hardware?
They were very rigorous. First, they used a GLM, a generalized linear model peer prediction algorithm. It looked at baseline firing correlations and actually predicted this would happen. Second, they built a massive computational DG circuit model. It confirmed this wasn't due to static synaptic weights, but a dynamic rebalancing of the network.
Dynamic rebalancing, meaning what exactly?
Meaning the brain is full of polysynaptic loops. Cell A talks to cell B, which talks to cell C, which talks back to cell A. So turning up the broad inhibition actually disinhibits a select few cells. By silencing the competitors, a few specific cells are suddenly free to fire like crazy.
Creating a winner-take-all environment.
Exactly. And to measure this, they use the Gini coefficient and the Lorenz curve.
Now, usually you only hear about the Gini coefficient in economics, measuring wealth inequality.
Right, but here they use it for firing rate inequality. Stimulating these interneurons significantly increased the Gini coefficient. The network became starkly unequal. A few rich cells fired continuously, while the poorer majority were completely silenced.
It's like a spotlight in a theater. If you want the audience to focus on one actor, you don't just shine a light on them under normal lighting. You plunge the rest of the stage into total darkness. By applying deep inhibition to the background, the one actor in the beam of light, the winning memory assembly, appears infinitely brighter. The interneurons don't just suppress, they actively select the winning memory by maximizing contrast.
That contrast is essential. Without it, your memories would just be a washed-out, blurry mess.
Okay, final mini recap. Bullet 1. Activating inhibitory cells paradoxically excites specific neurons. Bullet 2, this happens through complex polysynaptic network rebalancing. Bullet 3, this creates a winner-take-all dynamic, maximizing the contrast of specific memory signals.
Beautifully summarized.
So, listener, can you think of a time when blocking out all background distractions allowed one specific winner idea to finally break through in your mind? Let's bring this all together for the outro. We've seen that PV and SST cells have highly specific roles. SST gates the timing, while the whole inhibitory network dynamically creates this winner-take-all environment to select memory assemblies.
It's a self-regulating system.
Yeah.
But it raises a really provocative thought. We've seen that optimal memory formation requires this precise, unequal dynamic. But what happens in a human brain when that inhibitory balance is lost? Like in epilepsy or Alzheimer's.
Right. What happens when the nightclub loses its bouncers?
Do all the memories try to win at once, resulting in none of them surviving? Without PV cells absorbing the shock or SST cells acting as stop signs, the network becomes overwhelmed. The noise doesn't just drown out the signal destroys itself.
That is staggering. Well, thank you for joining us on this deep dive. For you listening, we'd love to hear your thoughts on that final provocative question. Do memories destroy themselves without inhibition? And what other cognitive mysteries do you want us to unpack next? Next time you find yourself locking onto a single voice at a noisy party, thank your PV and SST interneurons. They're working overtime, plunging the room into darkness, just so one voice can step into the spotlight.